Hello, welcome to Biology, the scientific study of life. Again, more about potential or membrane potential. Maintenance of membrane potential by ion bombs. All cells have voltages across their plasma membranes. Voltage is electrical potential energy, a separation of opposite charges. The cytoplasm of a cell is negative in charge compared to the extracellular fluid because of an unequal distribution of ions and cations on opposite sides of the membrane. The voltage across a membrane, called a membrane potential, ranges from about 50 to 2000 millivolts MF. The minus sign indicates that the inside of the cell is negative compared to the outside. The membrane potential acts like a battery, an energy source that affects the traffic of all charged substances across the membrane because the inside of the cells is negative compared to the outside. The membrane potential favors the passive transport of cations into the cell and anions out of the cell. Those two forces drive the diffusion of ions across a membrane: a chemical force, the ion's concentration gradient, and an electrical force, the effect of the membrane potential on the ion's movement. This combination of forces acting on an ion is called the electrochemical gradients. In the case of ions, we must refine out concept of passive transport. An ion does not simply diffuse down its concentration gradients, but diffuses down its electrochemical gradients. For example, the concentration of sodium ions Na+ inside a resting nerve cell is much lower than outside it. When the cell is stimulated, channels that facilitate Na+ diffusion open. Sodium ions then fall down their electrochemical gradients, driven by the concentration gradients of Na+ and by the attraction of cations on the negative side of the membrane. Some membrane proteins that actively transport ions contribute to the membrane potential. An example is the sodium-potassium pump. Notice that here passive transport and this one active transport. Substances diffuses spontaneously down their concentration gradients, crossing a membrane with no expenditure of energy by the cell. The rate of diffusion can be greatly increased by transport proteins in the membrane. Notice that active transport, some transport proteins act as pumps, moving substance across a membrane against their concentration gradients. Energy for this work is usually supplied by ATP. Notice that. That the pump does not translocate Na+ positive and K positive 1 for 1, but actually bumps 3 sodium ions out of the cell for every 2 potassium ions it bumps into the cell. With each crank of the pump, there is a net transfer of 1 positive charge from the cytoplasm to the extracellular fluid, a process that stores energy in the form of voltage. A transport protein that generates voltage across a membrane is called an electrogenic pump. The sodium-potassium pump seems to be the major electrogenic pump of animal cells. The main electrogenic pump of plants, fungi, and bacteria is a proton pump, which actively transport hydrogen ions, protons, out of the cell. The pumping of H positive transfers positive charge from the cytoplasm to the extracellular solution. By generating voltage cross membranes, electrogenic pumps store energy that can be tapped of cellular work, including a type of membrane traffic called cotransport. Cotransport, coupled transport by a membrane protein. A single ATP-powered pump that transports a specific solute can indirectly drive the active transport of several other solutes in a mechanism called cotransport. A substance that has been pumped across a membrane can do work as it moves back across the membrane by diffusion, analogous to water that has been pumped uphill, and performs work as it follows back down. Another specialized transport routine, a co-transporter separate from the pump, can couple the downhill diffusion of this substance to the uphill transport of a second substance against its own concentration gradients. For example, a plant cell uses the gradient of hydrogen ions generated by its proton pumps to drive the active transport of amino acids, sugars, and several other nutrients into the cell. One specific transport protein couples the return of hydrogen ions to the transport of sucrose into the cell. The proton can translocate sucrose into the cell against a concentration gradient, but only if the sucrose molecule travels in the company of a hydrogen ion. The hydrogen ion uses the common transport protein as an avenue to diffuse down the electrochemical gradient maintained by the proton pump. Plants use the mechanism of sucrose H positive co-transport to load sucrose produced by photosynthesis into specialized cells in the veins of leaves. The sugar can then be distributed by the vascular tissue of the plant to non-photosynthetic organs such as roots. Now, what we know about co-transport proteins, osmosis, and water balance in animal cells has helped us find more effective treatments for the hydrolation resulting from diarrhea, a serious problem in developing countries where intestinal parasites are prevalent. Patients are given a solution to drink containing a high concentration of glucose and salt. The solutes are taken up by transport proteins on the intestinal cell surface and passed through the cells into the blood. The resulting osmotic pressure causes a flow of water from the intestine through the intestinal cells into the blood, rehydrating the patient because of the specific proteins involved. Both the glucose and the sodium ion from salt must be present. The same principle underlies athletes' consumption of solute-rich solutions after a demanding athletic events. Okay, notice that this one, an electrogenic pump protein. Notice that proton pumps extracellular fluid, and this ATP cytoplasm H positive and H positive both. The main electrogenic pumps of plants, fungi, and bacteria are membrane proteins that store energy by generating voltage, which means charge separation across membranes using ATP for power. A proton pump translocates positive charge in the form of hydrogen ions. The voltage and Hg concentration gradients representing a dual energy source that can drive other processes such as the uptake of nutrients. Co-transport, active transport driven by concentration gradients. A special carrier protein such as this sucrose H positive. Notice that. H positive cotransporter is able to use the diffusion of H positive down its electrochemical gradients and into the cell to drive the uptake of sucrose. The H gradient is maintained by an ATP driven proton pump that concentrates H positive outside the cell, thus storing potential energy that can be used for active transport. In this case, the sucrose, thus ATP, is indirectly providing the energy necessary for co-transport. Okay, thanks for listening.